Good morning, everyone. It's a pleasure to be here. And I'm so sorry I couldn't be there in person, but I hope you'll get as much out of it as if I was there. Let me share my screen. So the talk today that I want to present is on our work on spectral graph model of brain oscillations. I'll focus on three themes. First, how do you fit this to empirical data? How do you explore its dynamics and stability? And then applications in neurological disease. I have no financial interest to declare. I want to start today by noting the obvious fact that one of the best ways to integrate the multiple modalities in neuroimaging that we see, MEG, EEG, fMRI, and diffusion MRI, is via a network analysis. And in particular, an emergent aspect in recent literature has been, how do you go from structural to functional connectivity imposed by these multiple modalities? And the key idea in this lecture is exactly that to explore these multimodal integrations, a graph theoretic approach is an excellent medium. But we need to go beyond statistical graph models and look at mathematical and computational graph models. And one key aspect in this lecture that I wish for you to take home with you is that simple linear network models can capture this structure-function relationship perhaps better than nonlinear or generative models. And of course, what I mean by functional connectivity, or FC, is nothing but the statistical correlation of signals from two regions of the brain. And anatomic or structure connectivity is the projection of fiber bundles between brain regions. Of course, the two are related, but not exactly and not congruently. So the rest of this talk will focus on how we can go from one to the other. To this audience, I don't need to describe the exact pipeline of neuroimaging processes through which we go from diffusion MRI and structural MRI to the network or the graph, but I would quickly note that what you end up with is a graph whose nodes correspond to regions of the brain and whose edges correspond to the amount of connectivity found to go from one region to another. So to begin with, I want to propose to this audience that a very simple linear model of diffusion on this graph is sufficient to recapitulate function connectivity. How do we model diffusion? Exactly as in the real physical space, where the diffusivity from region 1 to 2 is governed only by two things, the concentration gradient, x1 minus x2, and the fatness of the pipe, C12, and that's the part that comes from connectivity. So once you write that down between two regions, you can extrapolate to the whole brain using a vector value differential equation, which now involves the graph Laplacian, shown as L here. And the interesting thing about a network diffusion model like this is that the signal X, it could be functional activity or it could be something else, is given simply as a starting configuration X0 multiplied by a matrix exponentiation of the graph Laplacian. And this we can call the graph kernel or diffusion kernel, and so on. But the key idea in this approach is that the connectivity matrix imposed by this graph diffusion, that is C of F, is nothing but this graph kernel or graph exponentiation evaluated at a certain time t crit that can be empirically estimated. This was the subject of a paper we wrote in 2014 on a network diffusion model of brain activity. The idea here was that because the Laplacian is related to the connectivity matrix via a matrix exponentiation, you would typically do the exponentiation via a Eigen decomposition of the graph Laplacian, which means that the eigenvalues of the Laplacian are going to be the same as the eigenvalues of the functional connectivity matrix, but the eigenvectors are going to be the same, but the eigenvalues of the two are going to be related by an exponential decay. And that's exactly what we observe, as shown at the top left here. And of course, once you start with the main structure network shown on the bottom left and apply this eigenmode analysis with eigenvalues weighted by this exponential, you can recapitulate the mean functional network shown on the bottom right using just a small number of the eigenmodes of the structural Laplacian shown in the bottom middle. And as you can see, the more eigenmodes or eigenvectors of the Laplacian you add, the better your approximation becomes. But it is in fact sufficient to have only something like 8 or 10 of them to start looking very much like the mean function network. So what do these eigenmodes actually look like? Many years ago, we did a study on whether these eigenmodes are conserved between individuals, which they are, and they're conserved between atlases, which they also are, to a large extent. It starts kind of breaking down around 5th eigenmode or so, but until then, they are surprisingly congruous with each other. The idea of using Laplacian eigenmodes and eigenvectors is not really new. An entire field of graph signal processing has now emerged. And here I'm highlighting just a few of the earlier papers that refer to this aspect. And again, I want to point out that many extensions of this essential idea are now around, where, for instance, Abdel-Nour 2018 proposed an Eigen-mapping technique which expanded on the graph diffusion approach. Becker et al. Further expanded by adding a rotation matrix shown at the bottom of the screen. And obviously the beautiful work by Atasoy et al. Showed that a small number of these eigenmodes could recapitulate canonical resting state networks. So on that topic, I want to also contrast that with an approach by Abdel-Nour in 2018, who also showed that a small number of these structural Laplacian eigenvectors can actually explain the functional connectivities eigenvectors to a very large extent. So all of this combines to become what I call a spectral graph theory of functional MRI and functional connectivity. And I'm noting here some of the papers for the students in the audience who would like to pursue this aspect. This is an exploding field, but some of these earlier papers might be a really good starting point. So what new has happened in the last few years? The first thing I want to point out is that the Laplacian eigenmodes are great, but the Laplacian itself can benefit from more richer tunability. And a key aspect here is, and I'm reproducing the diffusion equation from the previous slide here, you see that if you replace the t, the time parameter, from a distant region coming into the current region with a delay term, tau 1, 2, and delays are after all realistic in brain networks because nothing travels at the speed of infinity. Now, if you add delays, in Fourier space, delays become phases, as shown here. And therefore, you can define a complex connectivity matrix where it has the magnitude is the same as the real connectivity matrix, but they all have now a phase between brain regions that comes from the delays introduced by signal transmission. So let's call that C star. And therefore, you can define analogously a complex Laplacian, which is basically coming from C star instead of C. And of course, these Laplacian and matrices have frequency dependency given by omega. In some work that omega is subsumed into the speed of transmission v via the wave number k. And this was described really nicely in a paper by Sear et al. In Neuroimage 2021. So what do these complex eigenmodes then look like? They look very much like their real counterparts, except that now, because you have these two new dials, alpha and k, you can dial them up and down for these eigenmodes, which used to be static, to now start giving you a lot of richness in space. as you can see here, the real Laplacian obviously cannot change, but the complex Laplacian can change its spatial pattern depending on how you dial these quantities up and down, which is really excellent because it allows us to then impart a very simple diffusive model with a very rich level of spatial information. And what do we gain with that? What we gain really is that if you look at the correlation between these, a graph diffusion model involving these complex Laplacians and various canonical networks, you see that the complex Laplacian uses far fewer eigenmodes shown in the x-axis at the top plot to get to a high correlation very quickly. And in some cases, it starts off higher and stays higher than the real Laplacian. Of course, both Laplacians are much better than random connectivity to achieve the correlations. One thing I would like to note is that the relationship between the ordering of these eigenmodes and the various canonical networks is not, in fact, very clear. Some people have reported that these low eigenmodes respond to functional networks and small eigenmodes do not. We haven't really quite found that. In fact, what we found is that certain eigenmodes, for instance, certain canonical networks, for instance, the default mode, encapsulate all the eigenmodes. Basically, every eigenmode is responsive to the default mode network, but only some eigenmodes are responsive to the visual network or the frontoparietal network or the somatomotor network. We think these are important aspects that require further study. So back to the question about the structure function map. Again, just to encapsulate that on a single slide, you can go from structure connectivity to function connectivity via any number of algorithms, all involving essentially the mapping of eigenvectors and eigenvalues from one to the other. And I've shown you some examples at the left plot here. In fact, a new version of this now tries to get joint eigenmodes, functional and Laplacian, together. And we showed recently that it gives better results than other competing algorithms, shown as JESM algorithm. This is very recent one. And again, as promised, here's a list of seminal papers in this area. The ones in green I've talked about. The ones in white I have not, but they're also very important and provide vital context. Even though the structure function models work well, they don't work too well. And one place that they fail is in explaining the interhemispheric function connectivity, of which you see a lot more, in fact, than the interhemispheric connectivity you see in structural networks. So it is no surprise that a structure function model applied on the Laplacian will typically fail in reproducing interhemispheric functional connectivity. So to address this issue, we hypothesize that perhaps there are latent interhemispheric connections between homologous regions in the structural graph. And I'm showing you the left plot here, what happens if you keep adding a little bit more and more of these latent connections from left to right. And you can see as you keep adding some weights, your model performance starts going higher and higher until it plateaus. So you don't want to add inordinate amounts of this connectivity, but a small amount helps tremendously. And the same is shown here in the matrices shown in heat map below. So that's well and good, but so far we only talk about function connectivity in terms of pure Pearson correlation. However, we know that fMRI data are autocorrelated, and so one ought to be able to look at the frequency signature of function connectivity as well. I think an aspect which has been really paid lip service to in our community so far, perhaps in the belief that there's really no frequency content in functional MRI. We don't think that's true, and I think a very simple way to analyze the frequency structure of functional MRI would be to take a rate model extending what we had before shown here, for instance. So it's a diffusion model as before, but now we have an additional term f of t, which tries to capture a cortical response function induced by signal entering in a particular region. We typically model that with a gamma-shaped function with a time constant. Once we do that, the model becomes imbued with tremendous amount of frequency structure, and you can write it as a closed form solution in Fourier space, shown at the bottom here. This is fantastic because what this tells us is that you can write the frequency response as nothing but a summation over eigenmodes, U. The eigenmodes represent spatial structures, and each eigenmode is weighted in frequency by a characteristic frequency response, which is governed by, in turn, the eigenvalues lambda k. This is a really nice separability between eigenmodes and eigenvectors in this, eigenvalues in this context. Some very simple, very preliminary results that I want to showcase here. This is work in progress, but we already find it that this spectrally resolved spectral graph model is able to produce the kinds of high bandwidth fMRI response going from 0 to 0.2 hertz, as well as the functional connectivity that we typically obtain. So a question here arises. We have seen in the community spectrally resolved DCM models before, which also seeks to create a frequency-dependent generative model of the bold response. And here I'm showing you, for convenience, the DCM signal equation, which is a multivariate autoregression process involving the matrix A, the matrix of effective connectivities. And again, you can write this in Fourier domain as a transfer function that relates the power spectral density of X to the power spectral density of the input noise V, which is usually assumed to be autocorrelated. So in the sense, both these approaches are trying to do the same thing, but this is where the similarities end. Because in spectral DCM, the goal is to estimate these matrix of effective connectivities, whereas in spectral graph model, we use a known matrix coming from the Laplacian and fit for global parameters that determine shape and of the spectral response. Therefore, we think of SGM as a structure function model and DCM as a model of effective connectivity. This brings me to the next part of my talk, where I want to talk about how you can do a spectral graph model that also accommodates higher frequencies, brain oscillations, especially those coming from MEG and EEG recordings. And this was the subject of a paper we published in Human Brain Mapping in 2020, whose goal was to kind of start with the work we presented so far and add more interesting delays and more interesting dynamics to it. And each node is now represented by a neural mass model of local neural assemblies. The neural mass model has an excitatory and inhibitory population. They have gain terms and time constants. And the macroscopic model is basically governed by the structure connectivity between nodes, in addition to a few global constants. First, the speed V, a global coupling constant alpha, and a global time constant tau G. And we want to take this model and try to fit it to frequency spectra coming from MEG. And this is the entire model. Obviously, as I mentioned, we need to introduce conduction speed. And as I mentioned before, there is a complex connectome now because delays become phase in frequency space. And then you can define a complex Laplacian, again, now as a function of frequency omega, shown here. Another thing noteworthy about this model is that there is a local response coming from the local neural mass model and a macroscopic response coming from this equation shown here. And both these responses are convolved with a gamma-shaped neural response function whose characteristic time constant is something that we need to infer. This is great because the equation can again be written in frequency space in closed form, pretty much as described in the previous work related to fMRI, where again the summation is over the eigenmodes U sub I, and each eigenmode has a spectral response given by the denominator here, which is governed by the eigenvalue. And this was the subject of two papers in OHBM 2020, NeuroImage 2022. And what we found is that the model is actually really good at capturing the spectral response of MEG data, both regionally, shown at the left, as well as at the aggregate levels shown at the bottom left. And in particular, it's actually better than a optimized neural mass model with coupling. And as you can see at the bottom right, the model can actually capture not just the spectral response, but also the spatial pattern induced, in this case, by alpha and beta band power. So question would arise now, given that both this SGM and previous neural mass models coupled to each other via connectome are trying to do the same thing with essentially a very similar math. In fact, the spectral graph math is linearized version of the neural mass model math. So what are the pros and cons? First, I want to note the similarities, which is that both cases use a node level local neural mass models connected via the connectome and solved by differential equations. You can simulate neural activity on the whole brain network, and both have now been shown to be proven in MEG, EEG, and fMRI. The differences are that in a nonlinear neural mass model with coupling, you have second-order nonlinear differential equations that require numerical integration to solve over long model times, and therefore the activity and function connectivity patterns can only be observed indirectly from these simulations. As a result, parameter inference is very tough, requires stepwise, manual, or heuristic optimization. Compare that to a linearized spectral graph model. It's a first-order differential equation, linear, has a closed form solution in Fourier domain, which is very rare in the field of computational modeling in neuroscience. In this case, the activity and FC are directly given by the solution. One thing I should point out that going to linear does not, in fact, seem to cause any loss of performance. In fact, the model performance of spectral graph models sometimes, quite frequently, in fact, better than coupled neural mass models. And perhaps the reason for that is that parameter inference is so simple and fast, and no hand selection is needed. You don't run into the pitfall of having gotten trapped in the wrong parameter space, which is typical in challenging inference problems like NMMs. So that's well and good, but what about stability and dynamics of a spectral graph model? So first, we want to look at the stability of the local or the mesoscopic model shown in the orange here. So we're talking about local neural assemblies with gains and time constants, and modulated by or stimulated by white noise. So we ran this experiment for stability, and because the model is closed form and frequency, it is not, in fact, easy to evaluate this in time for its stability. So what we ended up doing was we ended up doing an inverse Laplace transform, and we then modeled the stability of the inverse Laplace transform in terms of its eigenvalues and eigenvectors. And what we found was that you could easily determine a border between stable and unstable regimes of the excitatory and inhibitory populations within the local circuit. And we did this both with and without noise. And what we found, of course, is that the stability criteria doesn't change whether you add noise, which is obvious because it's a linear system, and therefore stability is a function strictly of model parameters, not of input noise. Compare that with nonlinear coupled neural mass models where metastability, multistability, and so on are governed, in fact, by noise. And it's a big aspect of neural mass models that the noise sculpts the stability. That is not going to be the case in a linear model like ours. Now going to the plots of frequency, where you can see by looking at you can evaluate the stability based on when the eigenvalues transit to the imaginary axis. And you can see, as shown in B, that there is a nice stable regime with respect to the gains and the time constants, where the time constants weakly modulate stability, but generally the gains are the ones that determine stability quite substantial. And you see in plot C that the frequency spectra in the stable regime can be shifted by the choice of tau E and tau I, the time constants of the two populations, as well as their gains. Now let's look at the macroscopic stability properties, where now again, we're looking for the long range connections and their coupling and their speed. So we did another set of simulations where now we allow every node to have the exact same local model parameters, which we then fit along with the other global macroscopic parameters. So what we found is that the two macroscopic parameters, alpha and tau G, have very strong and very well-defined stable and unstable regimes. Of course, for alpha, that's clear. As we know from previous work, alpha equals 1 forms the boundary of stability. Any alpha more than 1 in the kind of model we have will be unstable. That is not the case. It's not so easy to define a stable boundary for tau G, and yet it is very distinct coming from simulations. And in this stable boundary, you can achieve various modulations of frequency, peak frequency, depending on the choice of tau, G, and alpha that you choose. So the point here is that these two macroscopic model parameters are able to produce a wide variety and richness of spectral responses at different peaks of frequency, in this case shown at the alpha band, while staying in a stable regime. Just to point out, a detailed analysis of this stability and dynamics of this model are going to be presented at our poster 1732 later in this conference, so please check it out. Here's a result from that poster where we show that if you try to fit the spectral graph model in windows of time instead of the entire time duration of the scan, you see that all the model parameters, alpha, gains, and so on, are able to change over time. And in fact, alpha shows a very dramatic change from 0 to 1, close to 0 to close to 1 most of the time. And you can see it in plot E that leads to microscopic mesoscopic stability. Usually every brain is mesoscopically stable, meaning that the local model parameters are tuned to give you a stable response locally, but the macroscopic model is not. And this is the key thing. The stability of the macroscopic model in this particular case is governed very strongly by the coupling term alpha. It's very intriguing and is worth a second look and further study. But what we're finding is that about half the time, the brain decides it's not going to be stable. But of course, for a short period of time, and then it reverts back to stability. So that was all that theory working towards applications. So what are some applications of this approach? The key application is going to be in the case of neurodegenerative and neurodevelopmental disorders. So today I'm only going to highlight the neurodegenerative aspect, especially Alzheimer's disease, where just to back up a little, I want to point out why we want to do this. Because the spectral graph model is very parsimonious, it has only 6 biophysically interpretable global model parameters with no local variation unless you want to add them. It is able to fit to brain states and is able to fit to disease states. And if we're able to do this successfully, these global model parameters might then become biomarkers of disease. Now, this is important because the inference problem in this SGN world is so much simpler than in the neural mass model world, as I mentioned before. So we want to leverage that inference ease in order to apply this model as biomarker of disease. And many applications might arise: schizophrenia, autism, epilepsy, Alzheimer's. And of course, we can look for extensions in time-varying or dynamic models, which I will not have the time to mention today. But the general idea is that. So the first thing we wanted to do was to see if in Alzheimer's disease, we see the kind of shift in frequency that are captured by changes in the local circuitry. And I want to point out here that in Alzheimer's disease, there's a very strong downshift in alpha frequency band power as well as its frequency, and a very strong upshift in delta theta power. So to explore this, we first used the local only model with no connectivity. So these are just linearized neural mass models. And what we found was that in Alzheimer's disease, there's a dramatic increase in the excitatory time constant and a somewhat less dramatic increase in the inhibitory time constant. And they also come concomitantly with small increases in the excitatory gain and a small decrease in inhibitory gain. So this is really exciting because it allows us to look at the inhibitory excitatory balance, or imbalance, in fact, in Alzheimer's disease, an aspect that has been studied in mouse and in vitro models, but not really much in human data. So this is really exciting work that has now been accepted in eLife. I encourage you to check it out. So that's all well and good for the local model, but what about the macroscopic spectrograph model? After all, we're talking about the spectrograph model because it is able to integrate information through the connector. So what about that? So this work is ongoing, but what we have found by fitting a macroscopic SGM, keeping all local parameters uniform across the whole brain, with only 6 biophysical global parameters, what we actually, it looks like there are 7. What we find is that there's one parameter that really pops out as a difference between blue and yellow, Alzheimer's and healthy, and that is the network level time constant tau g, which shows a dramatic increase in Alzheimer's disease. There are other parameters that also change, for instance, the local excitatory time constant tau e, but others don't. For instance, the speed doesn't change, which might be a surprise to some people who think of Alzheimer's disease as having a demyelinating phase. We didn't find that. But the fact that these time constants change so dramatically is really exciting because this is kind of exactly what we expect tau and amyloid in the brain to do. And so to explore that, let's look at the plot on the right, which shows you the MEG between healthy and controls and Alzheimer's disease subjects, shown in A. And you can see there's a very distinct spectral shift in alpha band from lower to higher to lower frequencies. And the model is also able to recapitulate that as shown in the bottom of that plot A. But the way it is able to recapitulate that is by shifting the time constant of the network, tau g, from about 8 or 9 milliseconds to 18 or 20 milliseconds. So it's leading to a much slower neural response, and that's causing the model to shift everything, the alpha band to shift left, the delta theta to shift right, and to produce the right kind of spectrum across the entire wideband MEG frequencies from 0 to 45 hertz. I'm only showing you 35 hertz here. And to see if the shift in tau g has any relevance to the phenotype of the subject, we plot in C and D its relationship to CDR box score and MMSE, which are measures of cognition. And we find that there's, in fact, at least in MMSE, a really strong relationship. The longer your time constant, the worse your MMSE. We were limited by sample size here. In this study, I believe there are only 80 subjects. But we are really excited about this because this might be the first time you can see from whole brain MEG a distinct and specific biophysical thing that's changing, and that change causes the entire network to do something different in terms of its power spectrum. So this brings me to an end of my talk. I would like to thank my lab members as well as Dr. Sri Nagarajan's lab and collaborators. And I would like to end by saying that we are hiring. Please contact me for inquiries and job opportunities at ashish.raj@ucsf.edu. We have a lot of code on GitHub. So if you want to check out our GitHub page, have a look. We love to field inquiries, and we love to hear from people interested in working on this with us. Thank you for your time. Thank you for your attention.